It was 11:45 p.m. inside the university research laboratory. Carlos, a fifth-semester biomedical student, was exhausted after studying the central dogma all week. He was running a critical cell culture experiment. The cells were growing perfectly in their nutrient-rich growth medium inside the incubator. Carlos needed to isolate the mRNA molecule right after the transcription phase was completed. However, due to his extreme fatigue, Carlos forgot to wear his safety gear and accidentally sneezed near the open petri dish. "Oh no," he gasped. Contamination was his worst nightmare. He knew that foreign pathogens could introduce a random genetic mutation into his samples, altering the nucleotide sequence and producing a defective protein. If that happened, his entire semester project would be discarded. Panicking, Carlos quickly moved the sample under the sterile hood, applied a precise dose of antibiotics, and prayed that his biological catalysts would survive. Fortunately, the next morning, the DNA replication sequence was perfectly normal. Carlos learned a valuable lesson: never handle delicate genetic material when you are desperately in need of sleep.
Dr. Elena, a leading Mexican biomedical researcher, stood in front of a massive auditorium at an international science conference. She was there with the aim of presenting her team's latest breakthrough in cancer treatments. Her research focused on using CRISPR technology to perform precise gene silencing on specific tumor cells. Everything was going smoothly until a strict European professor raised his hand during the Q&A session. "Dr. Elena," the professor said, "your data shows that you successfully blocked gene expression, but how can you be certain that your technique did not cause unintended cuts in the patient's wider genome?" The room went silent. Dr. Elena took a deep breath. Thanks to her rigorous laboratory training, she answered confidently in fluent English. "Unlike traditional therapies, our guide RNA utilizes a highly specific anti-codon matching system. This ensures that our enzymes only target the exact nucleotide sequence of the tumor, completely avoiding dangerous cellular mutations." The auditorium erupted in applause. Dr. Elena had not only protected her scientific hypothesis, but she also proved that Mexican biomedical research is ready for the global stage.